if you ever need to look back. Okay, so the cilia and the flagella was the last thing that we left off on. One thing to keep in mind is that the cilia and flagella both have that 9+2 arrangement to the microtubules that build it, whereas centrioles that are involved in building centrioles in a cell, mostly you see those centrioles in the animal cells, they're going to be involved in, or I should say, they have a 9+0 structure, meaning they don't have any microtubules in the center. Those are kinds of questions that a lot of times you'll see on these biology tests, so I just wanted to make sure you're aware of that. First of all, know the difference, but second, know that these have that 9+2 structure. They're built of microtubules. Then they have those dining arms that pull on them, kind of like they're talking about with the arms and the monkey bars. Good way to remember that in that video when we ended that last time I saw you guys, which was on Tuesday. Okay, that should get us to the plant cells. And typically what you're going to see with the plant cells is, at least up close on the microscope, is you're going to be able to see some of the features of the wall. And that's what your book is interested in pointing out. So we have all these things on the inside of cells. We need to focus on the outer portions as well. So plants have cell walls, obviously. And so we just need to give some real brief descriptions. So the first one that's on your list is the primary cell wall. The primary cell wall contains lots of cellulose, and it's fairly flexible. So if you think about plants, and especially plant cells, they can expand and contract somewhat, even with that cell wall, to accommodate that vacuole that fills and empties. So you guys have all seen a plant that's wilted, I assume. Okay, those are made of cell walls too. It's not like it runs out of water and just holds its shape. It does wilt, it does change shape, and that's what the primary cell wall typically is like. It has flexibility that's built into it. Secondary cell walls will have put down cellulose at right angles to each other. In other words, the fibers are going to occur in such a way that you get a lot of strength. So if you have one running one way and run one in the other, okay, the right angles, got kind of the cross look to it, it's going to strengthen it up. Kind of like wire mesh. Just, the wire has squares pretty much to it from running the wires at angles that go across one another. And then the other thing that secondary cell wall has in it is something called lignin, L-I-G-N-I-N. So the secondary cell wall will have lignin. And plants like trees would have secondary cell walls. So on this photomicrograph right here, you have a plant cell. This is cell number 1, and this open space is the vacuole. And then cell number 2 is right here. The primary cell wall is going to be the outer portion of the cell wall. And then if they produce secondary cell walls, it's going to be found within here. And typically with those secondary cell walls, they can get pretty big. And you can see a big difference. The primary cell wall is right here. The secondary cell wall is in multiple layers at this point. And you'll see a lot of this forming things like trees. And if you think about a tree, even if it runs out of water, does it hold its shape? Yeah, right, the wood holds its shape. Last thing is this middle lamella right here. It occurs between two cells, two adjacent cells, or cells that are right next to each other. And this acts to hold them together, holds the cells, plant cells, together. A lot of time, this middle lamella will be pretty rich in a certain carbohydrate called pectin. Has anyone in here heard of pectin before? You can find it in the baking aisle. And what we've used it for in the past is to make jams and jellies. So you can take like fruit juice and a lot of sugar and pectin, heat it up to boiling, and then let it set, and it'll set up into a jelly. That's what it'll do. So you can make your own. Yep, there it is. Yep, that comes from cell walls. Yeah, the pectin. So you will see that from time to time, and this is where it comes from, the middle lamella. I shouldn't say the last thing, the second to last thing that they talk about towards the end of the chapter is the extracellular matrix in animal cells. And so there's a couple things that we should just make sure that you're aware of with the extracellular matrix. The matrix is going to be found outside of the animal cell in a tissue. And all these proteins that you see in here, so names like proteoglycan and fibronectin, collagen, these ones that are here, these proteins are going to be made by the cells that are going to be found near this extracellular matrix. Extra just means beyond the cell matrix. So why don't we go ahead and describe a few of those. And the first one I believe you have is integrin. Why don't you put as a protein in the cell membrane that connects to the extracellular matrix. and it doesn't seem like one that would probably be too hard to remember because it integrates into the cell membrane. So that's the word integrin, and I see integrate when I look at integrin. Collagen fibers, they're thick protein fibers that provide strength to a tissue. So for example, you have collagen in your skin. Okay, the skin has some strength to it. It's not just a collection of loose cells, it does have some strength. You guys are familiar with tendons, right? And ligaments. You guys had a chance to like see a real tendon or ligament? Like when you're butchering an animal or something? They're thick, aren't they? They're tough. They are packed full of collagen fibers, and that white appearance to them, definitely an indication that you have collagen. Cartilage, collagen. You're going to find all that in there. And then you have these proteoglycans that are also found in there, and to me they kind of look like fern leaves that are in here. So there's a protein with a lot of sugars attached. There's a lot of surface area that's here. Why don't you just put, they're sort of the fillers. They're the filler material of the extracellular matrix. So the extracellular matrix is going to have a very fluid nature to it. This is what's filling most of that space, or these proteoglycans. Now they can be involved in communication between the cell membrane and the extracellular matrix because everything's connected up here. I guess research-wise, they found that they can direct certain molecules to certain locations on the cell. So it's a little bit more than filler, but just know for now we're just going to call it filler. And then for the fibronectin, in terms of what its function is, it connects protein fibers together in the extracellular matrix. So the main thing I'm concerned about with the extracellular matrix, or just ECM is what your book abbreviates it, is know that outside of these tissues, remember tissues are made of similar cells with similar functions, you're going to have all these protein fibers that help hold the tissue together. Otherwise, it would be impossible to hold a series of cells together, if there wasn't some way to connect them. And that's what these different protein fibers that are secreted actually do. All right, the last thing we want to look at is the difference in junctions between the cells. So you have the extracellular matrix that helps hold a tissue together, excuse me, but you also have actual junctions between the cells. The first one's called a tight junction, and these are proteins that form a zipper-like pattern. So like a zipper that holds two sides of a jacket together, these hold two sides of a cell together pretty effectively. And the proteins are such between the two cell membranes that it forms a leak-proof barrier. So these particular cells right here, they're trying to illustrate, you see all these little finger-like projections coming up from them. I don't remember what those are. They look like flagella. That's actually a good guess, but it's not flagella. These are called microvilli, or villus is another name that's given to them. But there's these little finger projections that help increase the surface area of the cell, and it's mostly for absorption. This would be like the inside of a small intestine. You'll have these little finger-like projections that can absorb food molecules much more effectively than if this was just flat across the top. And then you have adhesion junctions, and I would just put they join two cells together but provide flexibility, so stretching. Okay, so these particular filaments allow for stretching to take place, and it's going to be in any organ or tissue that needs to stretch. So bladder stretch, stomach stretches, the heart is going to stretch and contract. And so you're going to find these kind of connections occurring between those cells. And then the very last one are these things called gap junctions. And what they do is they're proteins that form a tube or a tunnel between the adjacent cells. So they do join. You can see the proteins cross between two cell membranes. Zoom in on that so we can see it a little bit better. So these proteins that form sort of this round matrix right here, they do connect to one another, but the tunnels allow ions to flow between the cells. So the heart has to beat as a single unit, which means the ions have to be able to shuttle and move between the cells, and that's how they get through, is that right there. Plant cells also have connections. I didn't have this on there, but I just want to mention it. They'll have openings or gaps called plasmodesmata. Yeah, so they're right. They are right here. So a lot of times between them, the cytoplasm, or the cell membranes, I should say the cell membranes between the two adjacent cells, they'll join. Okay, and then it's capable of moving water between the cells, which is how you saw your onions shrink down, as water could flow between them based upon these plasmodesmata that existed. Okay, that ends chapter 4. I'm going to stop that. Okay, this is obviously a little bit shorter on the notes, which is good. But there's a lot of info in here that sets us up good for chapters 6 and 7. And the first one is this idea of entropy. It's a chemical concept. In chemistry, they hit this quite a bit as part of the second law of thermodynamics. But the whole idea is entropy increases over time, meaning that entropy, which is another word for disorganization, occurs spontaneously, and the amount of disorganization in the universe increases. So what that means is that at some point, and if whether you subscribe to it or not, if you think of like the Big Bang, that idea that everything started with an explosion and then all the elements were created pretty fast, and the stars since then have been cranking them out in pretty much their nuclear fusion chambers that they have, things are becoming more disorganized. In other words, energy is dissipating, molecules break down, and it takes energy to build them back up, and that becomes difficult to happen. And so entropy is usually characterized by bedrooms, because if you've noticed, how hard is it for your bedroom to become messy? How much work does it take? Not much, does it? You come home, throw your shoes over in the corner, toss your bag spills out, you don't really care, things fill out. And of course your parents are getting on you about it. And then finally on a Saturday afternoon, they've had enough, and then they make you clean it up. And then it takes hours to do it, and then it gets clean again. That takes energy in order to take something that's disordered and order it back up. And that's the way it works in the universe that way. Molecules are the same way. It takes energy to take single ones and build them into a more organized structure. And that's the key thing that the entropy is. In terms of how living things overcome entropy, because we're subject to it as well. Things break down. So what do living things have to do in order to build the molecules back? What do you need? Like every day, what do you have to have? Energy. You have to have food. Plants will use the sun. Some of that energy, of course, most of it's lost as heat as it works its way through the different levels of the food web and the food chain. But you have to have energy. So for number 2, how does life overcome entropy? By constantly taking in energy. Because if you're not taking in energy, either you're dead or you're probably going to be dead soon. You have to take in energy on a consistent basis because molecules are broken down and they need to be built back up, and that's what that energy does. Number 3, how is photosynthesis an example of life overcoming entropy? What happens in photosynthesis, you take carbon dioxide from the atmosphere, water from the soil, use a series of enzymes and other nutrients in order to produce glucose. So on the left-hand side of the diagram, you have 2 molecules, not very ordered, and then what do you build it into? This hexagon-shaped thing that has a lot of order within it. Now, anytime you build something more complex, you store energy in it. And it's the same thing with a brick wall like this school. There's a lot of energy stored in these brick walls. And how could you demonstrate that? What would happen if you could push it over? It's going to release its energy, right? And so you will see that. I mean, gravity plays a role in this as well, but it's highly ordered and it stores energy within it. It's a good way to think of just the brick wall. If that thing falls down and you happen to be standing on the other side of it, there's energy that's going to impact you. And it's a good way to think of it. So you go from less ordered to more ordered, which overcomes entropy. Okay, now one of the main molecules that this chapter focuses on, and it will actually for 6 and 7, is this molecule ATP. And there are 3 molecules that are involved in building an ATP molecule. First is a sugar. Okay, and the sugar happens to be ribose. And we've seen this. Ribose, deoxyribose. Ribose is typically found in RNA. So this carbohydrate right here, ribose, is one of the key components. The other one is adenine, which is a nitrogen base. This is the same adenine that's found in DNA. So you have ribose sugar, which is found in RNA, adenine that you find in DNA, or it could be RNA as well. And then if it was a nucleotide, the parts that make up DNA, RNA would have a single phosphate. However, how many do you see here? There's a PO4, 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 three phosphates. So there's 3 phosphates, a sugar, and a nitrogen base, which is adenine. Now, in order to make these things, anytime you attach on any chemical group to any molecule, it takes energy to do that. So there are these things called coupled reactions that oftentimes take place within cells. And the way that this works is if you have a molecule like glucose, it's highly ordered, it's storing a lot of energy. As you break it down, you can transfer some of that energy to ADP, which is the same molecule except one of the phosphates has been tore off from it, and you can attach the phosphate back on. So the idea of a coupled reaction is as you break down and release the energy of one molecule, you use that energy in order to store energy in another molecule. Coupled means that the reactions happen simultaneously and that the energy is released from one is used to store energy into the other one. So to better answer this question, give an example of ADP and PI combined to make ATP in a coupled reaction. Just tell me that glucose is broken down to release energy. That energy is used to build ATP. That's all I need out of that. So glucose is broken down, releasing its energy. That energy is used to build ATP. And the reason that we look at ATP, it's the energy molecule is used in almost every single reaction in a cell. This is the gasoline that powers the cell, if the cell was a car. That is how this thing functions. It's not diesel, it's not fast food. Right? It is a specific molecule that's going to run this thing. Number 6, give an example of ATP is converted to ADP in a coupled reaction. So this would be looking a little bit further down. So ATP is broken down to release its energy. That's what you can put for that one. So a phosphate is literally tore off of this molecule. And when ATP is formed, the majority of the energy is found between the second and third phosphate. This bond right here is a high energy bond. The others are much lower energy. Okay, so ATP is tore apart, releasing its energy, which can then be used, can then be used to build biological molecules. So this example says taking amino acids, the building blocks of proteins, and you use the energy from the release of ATP to organize them into a protein. So when a ribosome is building a protein, it doesn't just happen spontaneously. Energy has to be spent in order to make that process happen. So far, so good. Number 3, how's photosynthesis an example of life overcoming entropy? You take less ordered molecules and build them into something that is more ordered. So entropy would basically say that the order will decrease over time. So this will spontaneously break down, which it will. Life has to overcome that. In fact, there's an argument that's out there if you're ever interested saying life shouldn't even be possible because of entropy, because of the second law of thermodynamics. If things are getting more disordered in the universe, life shouldn't be existing. But on the contrary, it exists because life fights that entropy and moves the opposite direction. It's an interesting argument that has no really good, valid argument to it. But nonetheless, it's out there. You'll see it. Had a sophomore many years ago bring that up. I hadn't heard of that before, so it was something I've paid attention to ever since then. Number 7, we're going to look at how enzymes function, and they function in metabolic pathways, which look like this. So metabolic pathways, you're going to go through some that have a gazillion steps. And literally, sometimes like on biology classroom walls, I don't have one in here, they have a poster of like cellular respiration. And all it is arrows and lines and circles all over the thing. Like you can hardly tell like where one line ends and another one begins because there's so many pathways that are necessary in order to release the energy that's in your food and convert it to ATP. So a good way to think of metabolic pathways is very similar to if you were wanting to make ice. And this is some honors biology you might recall because this is where I got the ideas from this book. if you want to make ice cubes, it's going to require that you pour water into a tray. Okay, so those are two key components that you need. And then you got to put it into a cold environment like a freezer, and that's how you get ice cubes. Well, what would happen if you want to make ice cubes but you didn't have the water? No ice cubes, right? Even if you had the tray in the freezer. What if you had the water in the ice cubes but you didn't have the cold? It just sits. So the way that this pathway works is typically there are enzymes, which are proteins, that are going to cause a reaction to occur between A and B. And then to get from B to C, there's a completely different enzyme that's responsible for that. And then get from C to D, which is the end product, you have to have a different enzyme yet. You have to have each of those components working or you don't get from A to D. There's a series of paths that are necessary. So what oftentimes happens in disease is one of these enzymes is maybe mutated and it doesn't work. If you think of something like cyanide, which I'm sure everyone in here has heard of cyanide before, what does cyanide do? Kills you. And the way it kills you, it interferes with an enzyme in this whole process that shuts down your ability to make ATP. And you run out of energy and you essentially at the cellular level suffocate, even though you're still breathing in oxygen. It shuts off that final reaction that dumps electrons under the oxygen that you're breathing, and you can't produce energy. That is the importance of these enzymes on a metabolic pathway. Now, in terms of the way the enzymes on those metabolic pathways function, we got to look at a couple of parts here. Number 8, why do enzymes speed up or cause chemical reactions in cells? They force, I'm going to put a new term for you to write down here, they force substrates to react. A substrate is something an enzyme acts upon. So depending on what it is, there's a lot of substrates out there because there's a lot of enzymes. A substrate is simply the molecule it acts on. So it might force two molecules close enough together to where they react. Otherwise, if you have one of these just sitting in water, you're depending on them to bump in or collide with each other in order for a reaction to take place. You can speed up reactions by heating them up, but most organisms don't tolerate very high temperatures, so heat's not really an option. You have to have enzymes in order for them to function. Number 9, the active site on an enzyme is where the reaction takes place. Most enzymes will have very specific locations because the enzymes are proteins and they fold, but they have certain places within these folds that are specific to whatever it's in charge of processing. So usually when they're illustrated, they illustrate them like puzzle pieces where the pieces fit in, but that doesn't necessarily tell the whole story, but it's a good concept. In other words, the enzymes have different shapes to them, which means that not anything can be processed by this enzyme. It is dependent upon the shapes as they go through and function. There is a hypothesis that's out there, and this is number 10, called the induced fit model of enzyme function. What does induced mean? Like, let's say someone is pregnant, they say, I'm going in to be induced. Right, they're going to be hurried along, right? They're going to break the water and maybe give some, I don't remember what they give for sure now. Stimulates the release of oxytocin that starts the contractions, which eventually cause birth to happen. Induced means it's forced. And so that's kind of a way to think of it here. This induced fit model is going to be a little bit of a force that's taking place in order to make these reactants react.
Enzymes are proteins that speed up chemical reactions in the cell. A special region on the enzyme, called the active site, has a shape that fits with specific substrate molecules. An enzyme works by binding to one or more specific molecules called reactants or substrates. Binding occurs at the active site. The enzyme and substrates form an enzyme-substrate complex. The interactions between the substrates and the enzyme stresses or weakens some of the chemical bonds in the substrates. These stresses...
Well, you should have noticed in that little animation, you guys see the shape change down here? All right, so it's the idea that it kind of functions like a puzzle piece. However, since it's a protein and just a molecule in general, when you attach one molecule to another, both of them are going to change shape because you've changed some of the ways that these chemical bonds are going to interact. So you had positive and negative interactions going on that completely changes that, and the whole idea of it changing shape makes
encourage
full sense.
A link between the two substrates, leading to the formation of a different molecule. As a result of the chemical interactions within the active site, a new product is formed. The product is released from the active site, The enzyme assumes its original shape and is free to work again. Although this reaction has specifically illustrated the formation of a single product from two substrate molecules, other enzymes catalyze the formation of two products from a single substrate.
In other words, they break it apart, is what they can do too. So there's different reactions from chemistry. You probably remember single displacement, double displacement, synthesis, decomposition. Keep in mind, all those apply to this too. So it's not, I mean, here they show two things being forced to react together to make a single product, but it can be different, and it is different. So just one of those things that it all starts connecting up at this level. Number 11. How does the did you guys get 10 okay? All right, describe the induced fit hypothesis of enzyme function. What it basically says is that an enzyme and a substrate will both change shape and fit together. And it's not, remember, there's certain enzymes that function on certain molecules. And if I just pulled up like a random picture here, I should have did this earlier, of a biochemical pathway. Actually, let's look at glycolysis. That's something we're going to be talking about again here soon. Let's get a somewhat complex one like this one. It's not too bad though. All right, so here is the steps that are involved in breaking down glucose. So when you eat food and you have carbohydrates like sugars, they're eventually broken down by the digestive system into glucose, and the glycogen like animal starch, maybe in the liver. But each one of these with an ACE ending is a separate enzyme. So in order to go from glucose to glucose 6-phosphate, the enzyme that's required for that is called hexokinase. In order to go from glucose 6-phosphate to fructose 6-phosphate, you have phosphoglucose isomerase is the name of that enzyme. And then to go from fructose 6-phosphate to fructose 1,6-bisphosphate, you have phosphofructokinase 1. Each of these, different enzyme, because their shapes are slightly different. So you can't use the same enzyme for every reaction for the most part. There are a couple of examples, I suppose, where that does happen. I just couldn't tell you what they are right now, but that does happen. Keep in mind that most of these enzymes are very specific on the shape, which is why if you have single mutations like you do in Tay-Sachs disorder where fat builds up in the brain, because you don't have the enzyme to break that down, it kills the person who has that. Because that one enzyme is a super important enzyme that happens to carry out these reactions. And you'll see that. Sometimes one particular mutation in one particular enzyme, or if you have some kind of chemical that interferes with that enzyme's function, can be enough to shut a cell down. All right, so going to number 11, feedback inhibition. Enzymes will constantly crank out product if you let them. There's no brain that necessarily controls them, and so there has to be some kind of mechanism that exists to prevent enzymes from wasting all these energy and wasting all these resources, kicking out products that the cell or the organism doesn't need at that time. So for number 11, how does feedback inhibition prevent a cell from making too much or too little product in a biochemical pathway? I would put down the product controls how much of itself it makes. Okay, so the product controls how much of itself it makes. In order to explain that, we'll just use this animation.
Many of the enzyme-catalyzed reactions that occur in a cell, such as those involved in the biosynthesis of an amino acid, are carried out in a specific sequence called a biochemical pathway. In such pathways, the product of one reaction becomes the substrate for the next reaction. If the end product of a pathway, such as an amino acid, becomes available in the environment, it is unnecessary and wasteful for the cells to continue to produce the product. Cells therefore have the ability to shut down a pathway when it is not needed. In feedback inhibition, the end product of the pathway reacts with the first enzyme that is unique to the pathway. The reaction occurs at a site on the enzyme that is different from the active site, called the allosteric site. When the product binds to the allosteric site, the enzyme undergoes a conformational change and can no longer react with its substrate. There is no
Conformational means it's an overall shape change.
Substrate for subsequent steps in the pathway, and the final product is no longer synthesized.
Okay, so really, hopefully that explanation makes a little bit more sense now, that the product itself controls whether or not more is made. and that's feedback inhibition. Why is this important? Right, otherwise you waste energy. It reminds me, let's say like in the wintertime, you decide to open all your windows and doors and turn the heat on. Okay, and that's the way you're going to stay warm. What a waste of money, a waste of energy. You don't need to do that. There's more efficient ways to heat your home than that particular method. And that's kind of what feedback inhibition is. It's all a way of conserving energy. Which reminds me, did you guys see in the news this morning? Now you can argue with your parents. Laziness. Did you see there's a study on laziness? You'll like this. It's ingrained down to the cell level to conserve energy whenever you can. Laziness study. Let's see. It should be in the news. I saw it this morning. Marijuana use leads to laziness. It wasn't that study. New study proves that lazy people are more intelligent. Now that would be, who knows? From all the stereotypes I've seen of it, I don't know. Laziness. It was on ABC this morning. All right, so I typed in laziness study ABC, and I get a study on Hillary and a Donald Trump. I'll put the keywords that I heard this morning. Energy conservation. There we go. Apparently it took a while. This is in the Daily Mail in the UK. It's hitting us today. Why we are so lazy? Humans have evolved to conserve energy and avoid exertion. I mean, it comes down right to the cellular level. And so if you always wondered why you don't want to get anything done, you want to lay on the couch and just eat, you're trying to conserve energy is what you're trying to do. Only problem is now we live, in a particular environment where we don't necessarily have to conserve energy. If you're hungry, you can just go down. You can get in your car and drive to the fast food lane. Now you have to get out of your car and food is handed to you. You don't have to hunt it. You don't have to find it. You don't have to dig it up. So of course it's led to all sorts of issues, and trying to overcome those is tough to do. I thought it was kind of a fun study dealing with energy conservation that you can see right down to the cell level. You don't want to be wasteful, because if you're wasteful, it can end up costing you your life. See how many more we want to do here. Let's get through this enzyme stuff, and then we'll stop. Yeah, we'll probably get to number 12 and stop. This will be the last one we cover today. Energy of activation is the idea that in order to start reactions, it takes energy to do so. In other words, like the paper that's sitting on your desk that you're writing on right now is not going to just suddenly catch on fire unless we do what to it? Add energy, which we can do with a flame, which we could do with enough heat. Like if you put that in an oven at 400, even though you don't see flames, it'll be in flames in a short period of time. Energy of activation, your enzymes will lower how much energy it takes to start a reaction, which is why living things can carry out reactions at much lower temperatures than what could be done in a test tube or in a beaker or something like that. Okay, so that energy activation is how much energy it takes to start a reaction. And then number 13, actually number 12, we'll just call it good there. How do enzymes lower the energy of activation? They force molecules together. Why don't you just put they force molecules together to react, where otherwise you're dependent upon them to collide by pure chance out in the environment. Okay, we'll call that good.